Hi, my name is Vin. I'm a second year master's student at Columbia.
Hello, my name is Isaac Otari. I was one of the members of the team which worked on the project. Autoencoders are all single-cell data integration needs.
And I'm Erica, a master's in computer science student. Single-cell multi-omics sequencing enables the simultaneous measurement of cellular characteristics like RNA expression and protein abundance, which provide a deeper understanding of omics relationships. But datasets often contain overlapping modalities, which create a mosaic-like structure and makes integration challenging. Traditional methods often rely on overlapping features and often loses valuable information from non-overlapping areas. StabMap addresses this by incorporating both overlapping and non-overlapping features for more accurate cell characterization. And building on this, we propose three variational autoencoder-based pipelines to replace the PCA step in StabMap, minimizing data loss during dimensionality reduction.
The first method we implemented was an MLPVAE, or multi-layer perceptron variational autoencoder. This encodes our input matrices into the latent space and then decodes them using a Gaussian constraint, which is added to the encoder. This results in a mean vector and a standard deviation for our learned representation. Um, The MLPVAE architecture is simple to implement in Python using the PyTorch library. It utilizes fully connected neural networks for both the encoder and decoder portions, and is flexibly applicable to various data sets, and there's no need to change the underlying architecture. Um, Adding on, we utilized an MLPVAE as a baseline for how well our model could both handle missingness as well as integrate different modalities into a mosaic-like embedding. We utilize cross entropy as our loss function and utilize input normalization to handle large values and distinguish between zero gene expression and missing features.
The second method our team explored to try and build upon the MLP was to use transformers, in particular early fusion transformers inspired by computer vision and multimodal models in that field. The way this model works as follows. You take in n modalities and then normalize them. This is to make sure we handle large or small values as well as move zero values away from being zero. After that, we then multiply each of the inputs by their missingness mask, which is pre-calculated during data pre-processing. This in turn turns all missing values into zero while it's still retaining zero values as a non-zero number. From there, we can then do linear projections. And because the missing values are now zero, they won't be included in said linear projection. We can then concat the linear projection across all modalities and provide that into a transformer encoder block, which has several transformers. And then finally use another linear layer to collapse the larger vector into the desired output embedding size.
For our third method, we expanded on the second VAE method by mapping the transformer encoder's output embeddings into a curvature -1 hyperbolic latent space due to the hyperbolic space's ability to capture latent hierarchical structures. We computed the mean and log variance of the latent variables from the transformer encoder's output, and then we sampled from a standard normal distribution, prepended a zero to the sample, and then reinterpreted it in the tangent space at the origin. And this vector is then parallel transported to the tangent space of a learned mean parameter, followed by applying the exponential map to project it onto the hyperbolic manifold. This approach is called a wrapped normal distribution, and it enables our model to effectively capture and represent latent hierarchical relationships across multiple modalities.
So taking a look at our results, we find that all three of our models beat our StabMap baseline both in terms of Jaccard similarity and k-nearest neighbors using 5 neighbors. This is the same value that they used in the StabMap paper. We find that our transformer model performed the best, followed by our MLP-VAE, and then the worst performing one was a hyperbolic transformation. Although all three of these models perform quite similarly in terms of Jaccard similarity. The key finding was a clear separation in modality in both the training and test datasets for our transformer-based models. This highlights their ability to both differentiate between distinct cell types as well as effectively handle missingness. Um, We also examined different loss functions. We initially used a soft k-nearest neighbors to group cells based on cell type. It's inspired by the StabMap paper. However, we found that cross entropy worked best as it avoided collapsing our output logits into a single point. So to conclude, we found that our best model was the transformer model coupled with cross entropy loss. This provided the best performance in both cell type classification, not only achieving high accuracy, but it also was able to effectively handle missingness, as demonstrated in the PCA embedding shown below.
For our future steps, we can apply the proposed method to additional datasets to evaluate its generalization and robustness across different domains and different data. We can also explore alternative loss functions to further optimize our performance. And finally, we can investigate different clustering strategies for modalities, like evaluating similarity based on gene expression instead of cell type to gain new insights into the data's underlying structure. Thank you so much for watching.